folks will come on in and have a seat and we can continue with our session. So this afternoon, our last session is entitled The Way Forward. We've put a great deal into this day in terms of looking at what has gone on before, to get an idea of where we're at, what's required for a FANG type of project to be successful. And so now comes the part after these conversations, we can kind of share our collective ideas and figure out where we go from here. So okay, so we'll get started, and Chris asked me to let everyone know Jeff Silverstein was originally to be here and participate in this session, and he put a great deal of work into the FANG. He's one of the organizers of the workshop that was held at PAG a few years ago, and so really appreciate the effort that he put into it. And so I'm coming into this to step in and lead this session. And I think the first thing that we're going to want to do, we're going to ask Carl Schmidt, who was funded by NSF for the Research Coordination Network, to share with us some of the ideas that he's had behind that.
So this Research Coordination Network proposal came right out of the FANG discussions that were held at PAG. And so there's the objectives that went into the proposal are right here. And obviously you can read those. One of, I think, the major objectives will be to, it is, I hate to say this, largely funding to support meetings. So there's a couple of things we've already done, which is support postdoctoral fellow to come to this meeting, for example. But one of the main things that we want to do is try to bring together people with overlapping interests that can perhaps generate new grant proposals that make use of new collaborators. So one part of the objective is to support the efforts of FANG. And in that note, I'm actually hoping that we can have a meeting possibly next summer, where particularly we bring together groups directly interested in RNA-seq, and working towards methods, metadata collection, those types of things, so supporting the FANG efforts. And then the other, it turned out that coincidentally with this proposal, a group of people working on decapods submitted a very, very similar proposal. And Steve Ellis got us together and said, well, I can't fund both of you. And so we immediately shook hands and said, yeah, we'll take the money. So another objective of this is in many ways the decapod community, the shrimp, the lobsters, the crabs, etc., are maybe a year or two behind where this community is. So part of the objective will be to bring together people in this community that perhaps can put together grant proposals to help the decapod community. So the overall objective is to support FANG and also the decapod community. And I think that's really all I have to say at the moment. If any of you are willing to self-identify in terms of perhaps being a part of this research coordination network, I'd really appreciate that. And of course, I'm perfectly willing, as the few of you that know me, to hound you also into participating. Thank you for letting me share the RCN.
Does anyone have any questions for Carl about the RCN?
You have a meeting that's an organizational meeting for RCN. People could attend if they talk to you.
Yeah, of course, of course. And actually, our website is finally up and open, and there's a list for FANG, right? So I can just send that website out. Okay. So any interest will be great.
All right. Thank you. Thank you, Carl. So what we want to do now is kind of report out on our breakout sessions. We'll start with someone representing the data creation group. If you are prepared to report out. Joan, is that going to be okay? All right.
So there was a vigorous discussion of things within the data collection group. And most of us agree that there's a core set of 10 assays that need to be done. And many of those assays can be done in a variety of labs well. And then a few of them need very specialized labs. And how we do that, it was not fully decided. You want to add anything?
No, I think that's.
So.
That will be a follow-up on that.
And okay. Then the thing of having a set of reference tissues, there was a variety of suggestions from everything from 50 tissues from one animal. But then you have the issue of how fast and how do you collect all those tissues? And do we need to have that many tissues available for doing the basic core Fang assays? Would it be better, and this was not decided, to have 10 core tissues that are selected from 5 species and use those for the basics to do the assays? Then we can then expand out from that as those assays are developed.
But we will collect more tissues from those animals where we do at least the start of the 10 core tissues we select first.
And the suggestion was 2 males, 2 females, to have it so that you represent the genders and you can get the specific tissues that are relative to the genders.
About the tissue collection, we have to, for the tissue collection, we will make at least pictures will be taken from every sample. The location where the sample will be taken will be, we will try to match this over species as much as possible. So there will be a recommendation on how to do that.
So that's an important thing of the protocols that are on the Fang website. So if we want to say that in the kidney, you're going to collect a specific site in the kidney, that, you know, should be documented with pictures, but also trying to do that as we set up these comparisons to do that now so that we have a suggestion of what are the most important places for the different tissues. And then the problem of sample storage. Not every assay can do with -80 stored samples. We want to have multiple aliquots if we're going to have aliquots that are done for the reference and then shared with other labs. So we need to have that. But then for certain assays, there are ways to store the samples, whether it's RNA later or just snap freeze. So again, that's where the protocol section of the Fang website is important, that we have, when you're collecting the liver tissue, you're going to collect this many samples, you're going to collect multiple samples of this condition and multiple in another condition.
And you record the status of the individual which you will submit to biosamples on the status of this individual.
And one of the things is we shouldn't miss this time now to collect samples, because we have a lot of labs that are interested in doing this, but we want to have core sets for each of the species and have those available to go for a funding request for, you know, Graham Plastow suggested across multiple funding agencies. If we have an agreed upon core set of tissues that we can use, then we can potentially go to multiple funders and say, this is what we want to do. Let's pull those funds together.
I think that's more or less.
So. Huh? We're a good team.
We practice.
We debated the whole time the discussion was going on. Any
Question.
Ola?
So, when you say that.
So when you talk about the health of the animal, in what sense are you talking about health?
Yeah.
I mean, generally you will sample an animal as being healthy, or do you want to have some professional people to look at the animal to see if it was healthy?
Yeah, that's an important thing, is just to record in the bio data set what you know about the animal at the time. And we understand that, you know, what you might consider a healthy animal, what I might consider and the vet may consider might be different. But we're just saying this was done at this age, at this time, at this place. And for the reference, that should be sufficient. Now, we each have data sets that are much more complicated. And this is just getting from 4 animals for each species. You know, doesn't need to be hundreds of animals. That's what you'll do for your data. For Faang, we want to have this core set of material.
I wanted just to say that in response to Ole, yes, to assess if an animal is healthy is not straightforward. Could have a different definition depending if you have a veterinarian examining the animal or you're just guessing it, okay? So there are a lot of terms like this, which are actually very vague, which will go in biosamples. But the idea is that this essential information is what goes into biosamples, but then all the linked information is kept aside. Like there will be an institution which is carrying out a collection. And so there will be many more information that we don't overload biosamples with that, but it can be still retrieved after, once you have, right, Laura?
I would personally not advocate that solution because the odds of the host institutions being able to retrieve that data in 18 months, 2 years, 3 years, 4 years is very, very small. And so if there is data that is important, it should be in the biosamples record in the first place.
Thank you.
So,
And you can always add information later. You know, if you sequence an animal and you sequence 3 bacteria and 2 viruses, put that data in later.
that's the point. That's the point I was going to make. We're not very good at identifying animals that are sick. So we've just done an experiment looking at feed efficiency, high feed efficiency, low feed efficiency animals. And of course, the low feed efficiency animals we selected not because they were sick. We deliberately avoided trying to do that. But in the process, what we find when we look at the RNA-seq data is immune function genes are upregulated in those animals. So if we're doing RNA-seq on all of these animals, the RNA-seq data will tell us if these animals are actually sick or not.
Yeah, I will also look at the histology because sometimes we see like we don't see anything in an animal, but looking at histology you can find.
I'm sorry, what did you say?
Yeah, histology. Let's also look at the histology of the animal. So I think also that Red that store also the samples.
Yeah, we've stored samples both for freeze snapshots and paraffin embedded tissue.
Also for later on.
It's just one issue. You're talking about you're putting data into biosample, and but then more information may become available afterwards.
Right.
And so it's a question of who owns the biosample and who has the right to update it can be problematic, because usually it's the submitter, the original submitter of the data, who can make updates. But if a third party discovers useful information about the animal or the sample,
be added by the person who submitted the samples.
That may not be the case. You know, you have postdocs who leave and go on to another thing. It's you know, for NCBI data, you know, we've knocked on that door and said, this data is crap. but the original person won't change it, and NCBI doesn't have the rights to change it, we have to think about that. It's an issue. Laura wants to make a comment on that.
Use the microphone.
There's a mic right behind you, Laura.
From an update perspective, if Faang has a DCC, I know at the EBI people are keen to develop scenarios that allow brokering of metadata as well as brokering of actual data. So it might be we can come up with sensible solutions to this problem, but they do need to be thought about, and the solutions will need to be implemented because they don't currently exist.
With due respect, as a veterinarian, this concept of being sure that the animals that you are sampling are indeed healthy and that their tissues are not infected is very important. And there are actually people who are trained and experts in this, and they're veterinary pathologists who are boarded to look for disease, to look at the histology and be sure. Not that we can be perfect, but if we are looking for regulatory networks that are actually like homeostatic, then it's really important that these animals not have, and we're not talking about people who can tell you that they don't feel well or about mice that are kept in clean colonies. We're talking about animals that are outside on grass and they have bovine respiratory disease circulating through them, and they have parasites, and I just think that if we're talking about a very finite number of animals, and we're talking about investing the kind of money and effort that we are talking about in these pipelines and these outcomes, we really need to make just a reasonable investment to be sure that those are pristine individuals who are entering this effort.
Well, not necessarily pristine, but characterized.
Yeah.
I think I would use that, because you're never going to find an animal that doesn't have commensals or pathogens.
Sorry, just a question. For the reference samples that you're planning, are you planning on having a veterinarian there?
Well, I think the recommendation has just come that would be.
And also have them review the
samples
pathology slides.
You know, you collect the samples appropriately.
For example, in our case, we prepared section in paraffin for all the tissues, and we kept them. I don't know if it will be used one day or not, but that may be part of the answer. I mean, you could go to check at the histological level if you know what to look for. Otherwise, this variability, given the diversity of raising systems, yeah, exactly. Yeah, but my point is that if you have 20 data sets and then you try to cluster them somehow, these, let's say, sick animals should cluster away. Okay, so it's a kind of a objective way to be sure, not to be sure, I mean, to gain more information about that. You wanted to talk?
Another way to handle that is if you have access to, as we do, cattle that are in feedlots, and you measure their growth and you measure their feed intake and feed efficiency, and you select the animals that have the highest performance. Almost by definition, these are the most healthy animals in that population. You avoid all of the sickness.
Not all the sickness. Your microphone right behind you.
Just like the opposite of that, with chickens which had high feed efficiency, high breast muscle, and those were infected because they had muscle disease. So it should be checked either way, like if it is high performing or low performing, because in our case, it's just opposite of that. The high performing chickens are having.
I think healthy is a very difficult status. And I think you have to indicate as much as possible what you can, and not saying, okay, this animal is healthy, but we assume we tested this and this and this, and this was free of. And that's what we have to deal with, because otherwise you can't say this is absolutely free from all diseases. That's not possible. Yeah. Yeah. Laura.
Yeah, so I mean, humans obviously have different problems from a health perspective than animals, but there isn't. I imagine everybody in this room has some kind of health problem. But if you actually sampled all of us, we probably would all cluster together. So you can overthink this.
question.
I have a very general question. So what's going to be the use of this reference, and how is it going to be distributed, used, paid for? Or do we need it to move ahead, or do we need to wait for this? I mean, it's like a big thing. How are we going to see something with the reference and collecting the copyright from the essays, or?
No, as people said, do it now.
Yeah, start. That's the
And
most
people
important
have
thing. That's
done it
also what we heard in the sessions to start. So the thing is that we want to select the animals, and there will be material will be collected or is already collected. We will start with at least the 10 most important tissues, and if you have the possibility to do more, perfect, just do it. But we will compare the first analysis over species for those 10 different tissues which will be selected.
So
and
we
of
see
course,
those 10 tissues for the comparative across species analyses. You know, for within a species, obviously they're going to be very different things, and even within a species, dairy versus beef, you may have differences that you'll be doing.
and we hope that by this we have more or less summarized and the things, because we have a very difficult discussion on how to proceed, but I think we hope that it will be more clear or starting to become clear. Yeah, of course. Yeah, think big. Yeah, think big. Yeah.
So, as a member of the steering committee of FANG, I'd like to see if the ASA committee could make a recommendation by the end of the month, and we vote on it and get it done. So that the whole community understands at least what the level of requirements are now, so that when say move ahead, they're not sure exactly how to move ahead. So and you have probably 3/4 of us already done, and the recommendations already discussed. So I guess I'm just recommending or suggesting that you get a first pass done, and so that the FANG membership can agree to it, and then everyone knows what that first level is.
Let's thank our reporters. Okay, then. So we'll go on to our second group, the data analysis group, and who was our volunteer to report out for our other group? Was a sign. It was Stephen White as well. So
I don't have it, but
We have the slides that we discussed. Yeah, make slides.
You can try to find it. I'll see if I can find it.
okay.
Should be able to find it.
Go ahead.
I have it from you, right, Mick?
Sure. So we had kind of, as one would expect, several discussion points, and we went over several issues from multiple angles. But I'll try to compact some things, and please do jump in if I'm missing something. One of the questions was, do we want fuzzy or hard pipelines, and how are we going to handle those kinds of issues? There was encouragement to set a defined time and then don't go for perfection, do something, which again, repeated theme. There were questions about what does a hard pipeline mean? Specifically, does that imply that everybody's using, say, the same instruments for the same, so in order to have the same software that would accurately analyze all the different data? That was the consensus seemed to be that probably was not as big of a deal as for example, the RNA-seq analysis software probably made a bigger difference in which platform you did it on. And there was discussion and support in terms of the pipelines at possibly using virtual machine images to go and do that, because that way you could replicate across different computer platforms and other kind, and the exact instantiation of the particular software without having to worry that you got the wrong one at some critical point. So there was then additional discussion about some of the levels of data, and there was a proposed outline. Whether it would be good to group in two ways, whether the data were produced by Fang, whether the data were analyzed by Fang, and that leads to 4 kind of outcome conclusions. There was a lot of discussion about this and whether produced by Fang or not, and very much a desire to include all of the data that people have spent lots of time and energy to generate. And people were talking about distributing analysis products that have QC data and how to go back through and distinguish, okay, QC failed, QC passed. And also about the reference samples that meet certain quality standards. So you could go back and deliberately compare between laboratories and so forth. There were a few things that the discussion was that we probably are already actually doing pretty well. And so, for example, how to share the data, and so archives exist. How to provide access to the data. So hey, there's a private Dropbox, and so it's not fully laid out, but defining rules of use and go to Laura on some of these things. But at least hey, these would be positives. And there was positive discussion also about once the pipelines get firmed up a little bit more than they are, hey, would it be valuable to go and try to publish a paper specifically to say, hey, look, this is where we are, this is where we're headed. And generally, I think there was support for that. There were discussion about communicating with the other Fang committees and what needs to be said and is it being said. And so there was discussion of minimum standards, anything you can do with metadata, the more context you give, the more valuable it's going to be because you don't know what's going to be coming around the corner. Discussion of the use of ontology terms and whether to force, and I think the consensus was don't force but encourage as much as possible so that people could search in meaningful ways for the data that they needed. Specifically, one suggestion was that the QC process not refuse non-ontological terms, but give a specific message saying, hey, look, these are terms that are not in the ontology. Would you consider seeking these to be added? There was an interesting discussion about what could be done on stimulating collaboration. And the point was made about that there really is, in some ways, there's no reward for participation in some of the committees at this point. But that, hey, if people are spending a lot of time and energy going and doing this, and so they were talking about, okay, well, funding, and then the conversation turned into, wait a minute, there's a suggestion for, could we swap individuals between labs? For example, one group that's really good at this technique and someone else that wants to go learn the technique, and we specifically try to set up arrangements to do that, with the point that being that until people sit down with a common goal, that really a lot of things don't move forward like they could. There was additional support for grants and funding would be great. Also, some existing discussion about knowing what the gaps are and what's currently being done so that you can know how to fill it, and making these kinds of data available so you know with whom to collaborate, and then also so that grant reviewers can go and see, okay, this is how this fits, and trying to make it not just available internally, but even to those outside sources. The last section was on the translational impacts of Fang. There was a lot of echoing of our plenary speakers about predictive biology, about understanding the rules of life, using what is normal to help define what the disease states look like, and identifying the most powerful models for certain conditions, particularly models that don't have to be induced, that can be kind of a natural model. And the point being made that in a lot of the human drug pipelines, a lot of the drugs fail, and that arguably a lot of the failure could be, hey, we're using the wrong models to generate the drugs in the first place. And so if we could use regulome data or other pieces to identify the correct models, we might be really benefiting the drug pipelines, and that may be an argument that we could use to push things forward.
Yeah. So, I mean, just a few things I wanted to add, maybe as a global summary of all that was said, and you've been seeing on the presentation, is that I think we softly agreed on a few things. I mean, if we want to make conclusions of the outcomes of our discussion, I think we're going to go for hard pipelines. What that means is, if you have something to say, get involved now. These haven't been defined for any assay, any analysis. They will be defined, I'm hoping, within the next weeks, maybe 2-3 months. So get involved now, because that may be what's going to stick. I mean, that's going to be what you're going to be imposed in a way. In terms of data, we're going to, I think the idea was we're going to be inclusive. We're not going to, I mean, if data doesn't have all the characteristics we may be wanting from them to have in terms of QC, metadata, etc., we're going to take them, but that doesn't mean that that data is going to be considered in kind of global analysis. So we're going to try to include data, but highly characterize, or at least ask for people to provide as much information about it as possible. And probably when freezes come and global analysis needs to be done, there may be some thresholds that data will have to meet. And so bottom line is, for new data, try to follow guidelines as close as possible, because that's what's going to determine inclusive or not. And I have 1 minute, 2 more. So yeah, I mean, we need to communicate. And then collaboration, I think that, yeah, the idea was provide a reward for people working on it. And that may help everybody contribute and collaborate. And the impact, I think, was well.
So I think we have a question.
Yep. Pablo, can I ask a question about the thing you said just before that? In terms of trying to include all data. So one of the aspects of data samples is clearly the metadata. So how have you thought about, can people put in retrospective metadata? So in other words, data that was, or samples that may be useful, but perhaps not all of the metadata was collected the way you want it to be collected, because it wasn't listed before. So is that possible to retrospectively collect it?
Yes. So I think
or
there was a lot of discussion in this on whether we categorize data or not, and you had the different levels there. But my final take, and again, maybe this is a very soft conclusion, was that we're going to be inclusive of data that's people volunteers to the Faang effort, but then we're going to look at the quality of that data, the available amount of data, metadata provided, and then that may mean that that piece of data, that data set, may or may not be included in global integrative analysis. But, you know, because we don't know what those global analysis may look like, or who may want to perform different set of analysis or use a different bar in terms of what data sets can be included or excluded. I think it's better, we're better off just saying, you know, if you have information that you think is useful for Faang, we're welcome to list it in our, I mean, if there is an infrastructure. Or
I think data, all of the data in the archives, in the public, yeah, there we go. All publicly available data in the archives will end up being indexed by Faang. If it meets the metadata standards, it will be analyzed by Faang, and if it meets the analysis standards, it will end up being used by Faang in integrative analyses as appropriate. That is probably the solution, the best solution to take.
Yeah, that's a good way of summarising that. Write it down, Laura. I think, yeah, you know, we need standards for the global analysis when we write the Faang Nature paper. You know, we need to make sure that we're sure of the conclusions. So in one sense, we need to be elitist, but we don't want to say, you know, you can't be part of Faang because your data doesn't meet our standards. We need to be inclusive. That was the point. It's just having a framework that allows that.
Okay.
And I think I mean, just to add to that, I think we heard from our speaker last night that said, you know, just collect data. You never know how that may be useful down the line.
Can I just have one other comment on another point? Can you go forward? Yeah, there. So what do you need on funding and meetings and things like that? So the steering committee and the communications committee have discussed how to try to provide information on funding. So we will set up by the end of the month a place on the Fang website so people can enter data on what opportunities for granting agencies have put out for funding. And so that would be at least one place that people can go and search for funding opportunities of a particular type. And that's an evolving tool, but we're hoping that will be up in the next few weeks. So just a comment on that part.
I'm wondering if it's possible to run some pilot data on what's already in the public domain and what's already been generated within the consortium to address some of the questions about how the data should be generated. Because one question that was discussed but not resolved was whether there should be a small number of exemplary labs generating the data or whether every lab should be generating their own data. And of course, the concern with a catch-all approach is that you're going to be driving species differences which are actually a consequence of the way the data was generated rather than the biology of the animals. So it would be good to know how important that is if there's pilot data available to assess that now.
Yeah, I agree with. I don't know the answer to your question, but I agree with bullet point. Definitely, we need to avoid these kind of technical confounders of the biology. Jim had some.
Well, we did discuss that to some extent because we can go out, we could use human data today, which is a gold standard for the genome assembly, but go back in time to a less good genome for the human and say whether or not the pipelines, are they good for some species that may not have as high as quality a genome versus those that have something that is much better. So I think empirically, some of those things we could physically test, but we have to define this is that gold set that we want to try and compare these different points to. But it also would allow us to systematically test whether or not changing out this portion of any given pipeline is fundamentally better than what we had currently today. So it's multiple evolving system. Hopefully it gets better.
Any other questions or comments from our group? Julian, do we have any? Oh, actually.
So one of the things that we have been recommending is the data management plan within the proposal. So the data management plan is also, the consideration for the data analysis. So when you're putting a proposal, collecting the data, but you don't have a good plan for how you're going to archive the data, how you're going to process the data, and so forth. So that's very important, even just for, not only for the tools and resources program that I manage, but across the NIFA, I think for all the programs. So we are looking into it. I know the NSF is also very particular about the data management plan. So in this context also, I want you to think about also putting the resources for the bioinformatics. If you need to work on that, just not collecting the data, but like you should, if you need that the bioinformatic analysis, that should be part of the proposal. Just I want to make that. That's
So I guess we could come up with the data management plans that could be duplicated across many grant proposals, right? I mean, if we're participating in a FANG project and FANG has established, the appropriate data management plan, we can just make use of that rather than coming up with a whole new one if we can fit it right in.
Yeah, we have a data management plan, and Paul Fleechek, Laura Clark, Dave Burnt, myself pressed the button 25 hours ago to seek the funding to implement that with EBI, the Data Coordination Centre. And Laura will now explain what the data management plan is.
To be fair, the data management plan is kind of what we've already said about indexing all of the data in the archive. So you must submit your data to a public archive. If you do not submit your data to a public archive, it will not be part of FANG. And not only must you submit it, you must submit it without a holding to a publication date. It's not public, it's not FANG data. And at that point, yeah, ourselves or someone else who has the funding to run standard pipelines and produce standard distribution tools will be able to work something out. We have a lot of experience of doing this, as do some other people in the room. But we need to have the money to do it. And the more data that comes, I think hopefully we will get this seed money from BBSRC. But if we end up with 20 different projects on 10 different species and 70 tissues a species, some additional funds will be required because there is an overhead on a per experiment by experiment, per sort of project by project basis. So this will be able to get us a certain amount of the way, but it will not get us all the way if we are as successful as we hope to be in generating new projects.
Okay, any other questions or comments for this group?
Just on the funding, I think one of the things that we also discussed as a funders is there are existing mechanisms to do some of this work. I think that we're all appreciative of that, the work you put together to write the white paper. But along with that, like we see that there are some details that you're working on and so forth. But from my program, I can speak that the tools and reagents is very specifically designed for this kind of proposals coming in. But I think the important thing is going to be filling the gaps. Like whatever the proposals that are coming in, those are in line with what the community thinks, because that the tools and resources program, it specifically says that we are looking for proposals that are looking for community tools and resources. So this is the kind of thing that is there, but it's $500,000, but it's still, I think, for some of the work that you're trying to do, at least from the NIFA side, you have things, but if there are more plants that come up and if we see a need for that, so we might work on that going into the future. But at present, I think each one of you have to look at. I think NSF has also mentioned several programs that might be able to accommodate, depending on, again, like you have to talk to them, but it's having them, the international coordination piece that's there coming in and appearing, that's very important. I think as funders, like we see that it's a very important piece that would like to continue growing. And from our side, like I think we are also looking into ways that we could help the community support it in whatever way that we could coordinate. But again, each of us operated at different time cycles and all the ways that we need to satisfy our own, for U.S. Congress or whatever. So those are all the things that we work with. But again, we're appreciative of the community efforts and support. Anything you want to add, Steve, or something?
Thank you. I wanted to give a chance for our folks who are participating online, if they had a question for either one of our groups, please let us know. No questions online. Okay, well then, with that, let's thank our reporters. So our next speaker then will be Jim Reese, who's been asked to
there's a couple others.
We're going to give him a minute to load up here, but he will be kind of summarizing where we're at in the talk, ideas taken, and our next step summary. And I think Laura will be helping him out.
Did you send me to? You sent both of them?
Yes.
Okay.
It's all
and what about Jesus?
He's just talking. So just open one.
You have them all in one?
Yeah, I just incorporated it.
Big? All your
It's
damn
a little-
pictures.
Yes. Yes, for yours. So, we have the, oh, that's what we have to do. Okay. So before we get started in terms of doing this, I wanted to say a great thank you to everybody that is here and your willingness to put ideas out there for others to either agree with or disagree. Things like this will allow us as a community to do things that otherwise no individual lab can do what we're attempting to do here. And only by coming together and providing ideas and adding this or taking away from that will we move forward. So for that, thank you very, very much in terms of the time in which you've done so far. With that, I think we'll start with Jizu. So come and use the.
Thanks a lot. Give me this opportunity, and actually from our discussion, I really think everyone have wonderful input for these discussions. So from feeling also our work and also I have some suggestions and I want to summarize this kind of work. Okay, so for our discussion, what I think is we discuss a lot about how to collect samples and how to prepare reference animals and how to organize how many samples, tissues from each animal. Actually, we discuss a lot about that. That's good. That's really very, very important. And then for data analysis, and so we discuss about how to do the data quality checking and then pipeline preparation and the data analysis. All these are very, very important. But I think is for our purpose is mainly for functional annotation. So right now we seems ignore some part of that. Okay, so I think that's important because we are not there yet. So people didn't mention this kind of the idea and analysis strategy, this kind of stuff. So I think before we go to that step, when we prepare our sample and also include pipeline analysis, we should think about that. Okay, so for example, we collect the different, doesn't matter 15 samples or 50 samples or 100 samples. Okay, I think it's impossible for one animal in one lab to do 100 samples. Okay, 100 tissues. So that's costs a lot of money. I didn't know any funding agency can fund that kind of a project. Okay, but it doesn't matter for how many tissues, even for 10 tissues or 15 tissues or 20 tissues, already give us enough resources to explore useful information. For example, RNA-seq, what I think is not only to identify, for example, for the treatment, if involve any treatments, we can identify a significant gene list, and we also can do splicing analysis, intra-rotation, okay, and all of these things we can do that. And for other chip-seq, for example, histone modifications, and also is in this kind of stuff, even for DNA-seq, is very, very important. And for our livestock animal field, actually, I think from worldwide, and we have rich resources for GWAS, for example, this kind of SNP data, and we are there. And so we can combine this kind of SNP data, GWAS data with our, this kind of omic chip-seq data, okay, even methylation data. And I think another important thing, even we do few samples, we cannot do many samples for this. even do few samples, but we do many tissues. We also can do trends analysis. For example, DNA-seq with RNA-seq, combine that together to do sense or trends analysis. And we also can do sense or trends other histomarkers analysis. For all of these things are very, very important. Doesn't matter for which kind of conditions, for the pathogen exposure, and also for example, our group will get to grass-fed, grain-fed. And actually, we have this kind of data, and we explore this kind of functional analysis. I think this kind of thing is functional analysis. It's not just, oh, we do few some. That's a discriminative analysis, so that's not function. So if we do function, we do need involve treatments. That's what I think. And I think if we do analysis, prepare this kind of project, we will have really good hypothesis, and then you have good probability to get funding. Okay. Otherwise, just if that's general data, it's hard. That's what I think.
So we discussed this a bit beforehand, and I have just 3 slides that are mostly focused on funding. So we were sort of commended on our original paper, and the white paper is a great starting point, but we still need to expand our vision and to actually define what is our pitch, what are the end games here, and I think we were given wonderful information during the funders talks about the sorts of opportunities that were available to us and how we should present ourselves to the funding agencies. And yeah, I mean, this was the most succinct sort of description of how to move forward. We need a clear vision and ambitious goals. We need to prove whether there's an economic impact for the funding agencies for who that is relevant. We need to make sure that there is complete stakeholder buy-in, and that's from not just everybody in this room, but from the wider community and also from industry. And so it's really important that we work together to actually look at our funding opportunities and make sure we do things in a sort of consistent and coordinated way, rather than 20 different scattershot projects that do subtly different sets of assays and subtly different ways of sampling. Because in the end, while that is all useful data, it will not be as valuable when drawn together as if we do things in a coherent manner. And then finally, of course, the end of the funders talks was from Jean-Charles talking about setting up an international research consortium. And we really, over the next few months, need to consider how we would set up an international research consortium and what the goals of that consortium would be. I mean, it's really important to have concrete outcomes for these things. If you look at the sort of ERDIC or IHEC or the International Cancer Genome Consortium, they all have quite clear goals. And we need to make sure that we can define what our ultimate goals are and explain, both in the context of basic biology and in the context of sort of translational economic output, why those goals are important. Because otherwise, we will either exclude funders or we will sort of lose our way when it comes to actually getting something truly worthwhile out of what FANG is trying to achieve. I mean, at the moment, when I talk to people about FANG, I describe us as a coalition of the willing, because that's generally what we are at the moment. There's a small amount of funding, but there isn't a large amount of funding. And while it's wonderful that ENCODE got all the money it did, we can't expect to get $100 million to do what we've done, what they've done already. So we have to figure out how to do this with the smaller projects and how to draw the smaller projects together to sort of gain us value. And this may be one of the advantages of the IRC is, while it's a gentleman's agreement between funders, there are several funders that have sort of signed at least something to say that they recognize how important this is and they are willing to support moving it forward. And I think that's very useful. And I'm sure all of the funders in the room will agree with me when I say, when funding agencies tend to like it when other funding agencies believe the same thing is important as each other, it makes everybody feel better about it. So, but yeah, so that's sort of an overview from a funding perspective.
It changed things a little. So the community as a whole, we have grown in different steps. Almost everybody in here has played some role on one of the projects in which is here for the genome assemblies that are there. We had a good start. We're moving on to bigger and better assemblies for what's there. fundamentally, it has allowed us to grow together, to start to do things in a scale that prior to this probably really didn't exist. Correct, Alan? He's nodding his head. And by the way, Alan hasn't tweeted in almost 4 hours. I don't know what's wrong with him. Now, things are going to ramp up. I mean, there's 10,000 plus genomes coming. Now, that's beyond the scope of what we've discussed here in terms of doing. But the things in which we're trying to put in place can be used for many, many other organisms that will come along in time, such that I see that what we have is extremely important in that respect. But in doing this, it will also help us to come together in a different way. It's going to involve other groups than what we've had traditionally, because we need new expertise. We have to raise the standard in which we are physically working with. Now, that has occurred in other projects that have come along after the genome sequencing projects, whether it's the resequencing project for cattle, which, I mean, in the project, there's what, 1,600 animals that are publicly in that consortium. There's probably that much more that has been sequenced that's not part of it, but that has allowed the community to grow even more. The genome resequencing of multiple species has brought other groups together. Each one of these is learning fits and bounds in terms of moving forward. Now, as a community, we do a great job of collecting lots and lots of data on many different things, but it might not necessarily have been in the past in a very cohesive manner. We may have had a project that was just designed to look at transcriptomics of this viral infection. But as we're coming together, more and more technologies are coming forward. We have the ability to integrate them to a greater extent. What we're talking about here for the last day and a half is essentially exactly that. They are other phenomes that we're pulling together in order to describe that biology. Pablo said one of the things in which he liked the greatest is we're trying to define the rules of life. We got the data in terms of the book for the genome. Now, how are we going to put those rules out there as to how it's physically working? Now, in looking forward, in trying to do some of this, we have to have what are the impacts that we will move forward. Well, some of what we're going to do is just leverage prior funding or what's there. We don't have to go relearn everything that has occurred in the ENCODE project. Let's take what is appropriate there, the tools that are there, and use them for what we have. But then there will be new things that we'll have to add to it. Why should we go take and reassemble all of the database? We don't have to. These are not all of the ones in which we'll work with, but there are information there that we need to partner with where appropriate and will help us move forward in a manner in which we'd like to do. Now, compute, it's here, it's there. We're going to have to learn as a community, and some folks already have, that what we do will probably not occur at our institution. Frankly, I don't care where the computer is, as long as I got access to it and I can burn as many cycles as I need to burn in order to ask the questions I want. But at the same time, we can't just burn cycles non-discriminately. We are talking about, if we can do this correctly, of elevating the level of science and in essence, lower the times in which we have to redo the exact same thing over and over and over again. So how do we integrate what it is that we have, whether it's in the sample collection or the data analysis or the stories in which will come out from the biology and other things that are there? This, to me, is really important. We get this right, and we will have moved forward in terms of what we have. A personal vision of which I would really love, and this goes back to data reuse and facilitation, is that someone's going to generate some data somewhere. We're going to have pipelines and other standards in which we have set here. We'll have pieces, parts we're going to pull out, and we'll stick something else in because it's better. Fine, but we'll grow together. And ultimately, hopefully, we end up getting some resource that we can all drink from and utilize in order to answer the questions, because we have the metadata, we have the phenotype to pull together. But I don't have to go back and reanalyze something that Carl has already run through this and is now available to me, and I can stream just the quantitative portions of it, such that if I want to look at this one little piece of the genome, it's there, and I can take that slice and utilize it to a greater extent. Now, this is not simple. I mean, all of us have done experiments where we have plenty of metadata. We just never made it available to the rest of the world. It's in some lab book somewhere. So we're going to have to move forward as a community to do this. But it will come with fits and bounds. Fine. We'll be able to figure this out and go forward. Now, one of the papers in which I was introduced to as a graduate student, which frankly was published before I was born, is as applicable today as the day in which it was published. And if you haven't read this paper, go do it. Chaos in the Brickyard. We're not just producing bricks for the sake of producing bricks. We're making these bricks because we want to grow something that is bigger and better than what we have today. And there are elements in science that have moved forward in absolutely phenomenal ways. So back at the time in which this occurred, you were talking about, in essence, the birth of molecular biology. It was far outpacing many other forms of science that were there. I think from a genomic standpoint, we're really at the same point. We can do things today faster, better than what we had done in the past. Now, in what we are generating, to me, we don't have these edifices available to us today. Many of the things in which we've talked about, and which have loved the discussions that have occurred, we're talking about laying the foundation of what's here. Whether that is in the healthy versus, well, this might be semi-sick animal. These are great questions to define, but they are only the base of where we start. My vision in terms of where we're going, and I hope that will occur, is that people will continue to add additional conditions, additional ages, and other things to allow us to ask higher order questions that individual research projects today cannot physically allow us. I would love for a day that I could go to a database in some manner and say, what is the difference between these 5 different families of viruses and what makes them unique? What's common across all of them? Trying to do that in a single defined experiment today really can't be done. But we're headed in that direction and defining the standards, the pipelines, the other things that will allow us to get there. Now, to get there, it's much like sequencing costs. We're gonna have to have orders of magnitude decrease in the cost of either phenotype collection, phenotype being transcriptome, ChIP-seq, all of this. Because I would love to do this on populations of populations. Think about it. I mean, we can look at the genetics involved in binding of a given transcription factor. Today, I couldn't afford to submit a project. The funding agencies would laugh at me. But if we change technology and other things that will allow us to do this, it would be great. And we could eventually produce some larger edifice in terms of doing this and explain biology in ways today that any of us couldn't hardly fathom in terms of doing it. Now, anyone got an idea what this is about? Nobody?
Termites.
Termites. Oh, that's a great idea, but no, not quite. You, what? Epicite? I don't know. In terms of doing this, this is going in circles. Yes, it has. Some of them got a little twist to it. Copernicus. The change in which we had from going around the Earth, going around the sun. To some extent, I view this in the discussions in which we've done. We've got a leap forward in which we had essentially begun to lay that foundation to allow us to do that. Predictive biology, it was brought forward earlier. To me, this would be great. I would love to be able to predict that this is what is going to happen to this set of animals, whether it's for disease, growth, or whatever. And now I can perturb the system and try to predict what it was and say what I can do better. If I could, or any of us could do this, it will have phenomenal outcomes, not just for animal agriculture, but biology as a whole. We've done this as a community. For a few genetics folks will understand this equation. And then there is another step forward in terms of bringing in genotype to try and do the same thing. We're moving towards predictive biology. We will have leaps forward in terms of doing it. Now, as we get that predictive biology, we're going to be able to go after those hairy, audacious goals. How are we going to feed the rest of the world? I mean, to me personally, this is huge. I hate going to countries not as developed, in which the one I happen to be blessed in being born. Seeing young kids with distended bellies just drives you nuts. These are things in which we can take this foundation and address. Or at the same point, I go back to my own country and say, there's also biomedical models that are there for obesity, asthma, anything else. The things in which we're talking about will have applications to them. Then there's also evolution in terms of doing it. Martine said this is probably not the best depiction of speciation within the Suis family, which is fine. We'll find that out. We'll figure that out. But we'll also go back to the origin of the species and say, here's what happened for the different beaks that were there, for the birds in terms of the evolution that was there. But we also can't forget that we have, as humans, a long interaction. Martine also pointed out, we probably have one of the longest experiments that has been going on, called domestication. We've been doing it for 10,000 years. We're only now beginning to get a better understanding of what actually has occurred because of that. And we'll go back and we'll understand, hopefully, genotype, phenotype, environment interactions. Interestingly, there's a few different environments that are out there. Some of them are lush, some of them not so much. There's a few different breeds, even different species in terms of doing this. We as a community have a lot to be proud of and should tout in terms of saying why we have the better model in terms of doing it. Instead of being, well, we might be able to do this. No, we have, it's out there. The data in which John was talking about last night. We have the information. We just have to go out in order to say, yes, this does this. And now we can take it to NIH, to NSF, to other places in terms of saying, here's where what we have to provide and make it the most appropriate. This perturbation and interaction of species and treatments and time and age will only allow this data set to grow and only allow us to better define what is the biology that is out there, that has each of our own little interests. Carl Schmidt wants to understand what makes a chicken versus a cat. Well, we said 2 legs, 4 legs. There's a lot of, everybody has a slightly different portion. But we can bring all of that information together and define things into a greater extent than what we've been able to do today. Now, at the same point, as animal science and other disciplines, we can pull interactions together. I really hope that we do this correctly and we bring in all of the reproductive physiologists. We bring in the nutritionists. We bring in the health officials. We bring in the folks in meat science, whether or behavior. Who's ever had their dog just destroy something within their house when they came home? Or in my case, my lovely dog, she is a dumpster diver. She loves to go after the trash. There are unique aspects of what we have and other disciplines which we can pull in, and we are uniquely positioned in order to help do that. And as I look at it, a project like Faang is the framework to allow this, and it will continue to give and give as a project beyond what we can envision today going forward. And hopefully, we're starting maybe over here somewhere, and we'll end up having something that is much greater and grander than what any of us envisioned when we first started on this project, no matter how many decades ago that we got into research in terms of doing stuff. And hopefully that will semi-reflect some of the discussions and ideas in which folks have had over the last day and a half as part of the meeting.
Okay, thank you all very much. Do we have any questions or comments?
That was great, Jim. The title for this section is the next steps. Yeah, the next steps, and that was a great sort of vision, sort of big picture thing. I think possibly we're guilty as a group of not looking at small steps and not looking. You talked about the chaotic bricks and so on. We mutter that we don't have enough money to do this project. But I'm not sure we've actually worked out how much money we might need. I've said innumerable times without any evidence whatsoever that the ENCODE project cost 250 million to date, and we could probably do it for less than 1/10 of the cost per species. But I haven't actually sat down and done the calculation. Most of us, when we write, would the funders please put their fingers in their ears? Most of us, when we write a grant application, think, okay, well, to do this, whatever this is, we need a body and $20,000 or $30,000 a year to keep that body gainfully occupied. And sometimes when we break down the cost and the consumables, we attempt to fudge it and make it look as if we actually costed this thing bottom up. To be honest, we seldom do, but you didn't hear me say that. So do we know how many bricks we need to build this thing, and do we know how much a brick costs? So by how many bricks we need, then I think we're, from early discussions, we're talking maybe 100 tissues for individuals. And so this is per species. And what's a brick? Well, a brick might be those 10 assays. Mm-hmm. Do we know what the cost of those 10 assays on one sample is? We should have that number. Now, we need that number in a spreadsheet where you can change it as the cost of sequencing drops. Mm-hmm. And as the cost of sequencing drops, don't get overexcited and think the cost of doing that assay is going to drop in proportion, because there's the fixed cost, there's the library prep, but possibly more importantly, it's the bit before you've got the library. It's from the sample to the library. So it's doing the immunoprecipitations or whatever, that bit. So we need those unit costs, and damn it, we should go out and figure out what those unit costs are and have it in a spreadsheet and alter it so that we can go to the funder and say, okay, this is the unit costs, and are you good for 100 units? Are you good for 1,000 units or whatever it is? Mm-hmm. And possibly we should build things up that way. During the interesting discussion on data generation and samples, I think it was Hans Cheng at one point was making the point that in terms of passing samples around different labs that could do assays, he said he wasn't really sure he could have a student in his lab receive samples from someone else and run the assay in his lab. And I thought, hang on a minute, this project's actually different from your sort of one man and a dog, or sort of one person and a dog postdoc or student project. This one, there's a factory element to this. And frankly, we should not be using students to generate the data. There is a wealth of stuff for students to do inside this project, but it's at the other end where we've got we're going to be under-resourced, and that's the analysis. So we need to generate the data in a factory setup, not using students, using technicians. And if you're doing it using that model, then arguably having a few centers who are generating a particular data type becomes more sensible. And in terms of the funders, just to sort of reflect back, because I was born in 1963, so I remember a lot more than some of you. And I remember some of the origins of the Human Genome Project. And in terms of, we didn't have the technology then to do a genome. And John Solston in particular said, okay, well, let's start doing one cosmid at a time in C. elegans. So that was kind of take small steps. Another thing about the Human Genome Project was when they were trying to get the money together, I think it's true to say both NIH and the Medical Research Council in the UK were sitting on their hands. And Mike Morgan, I believe it was, from the Wellcome Trust, in essence, slammed his checkbook down on the table. We're good for 6 million. What about the rest of you? And it broke the logjam. So if there's any funder in the room who would like to repeat that Mike Morgan act, please step forward now.
Something I just want to say, I think we need to sort of recapitulating what Paul said earlier. We need to figure out what makes this community special and interesting. But we also we don't have to do every single assay that ENCODE has already done. We should be able to look at the assays that have been done and work out actually what we really need to do is good both total RNA and short RNA-seq, a couple of chromatin style assays and some kind of methylation assay, and pick the right ones and do those. And do those on as many different tissues in as many different species as possible, rather than picking a single animal or a single tissue and going really, really deep before we actually figure out, but wait a minute, we only really needed the first 5. And that's something that we can do with existing data, and we should be figuring that out empirically, rather than putting a finger in the air and going, maybe actually these histone modifications and RNA-seq and reduced representation methylation will be fine. We should check. That information is there. That data is already in the public domain.
Isn't that where we are with the genome biology paper?
Well, no, so it's worth noting that the assays picked by IHEC were picked not necessarily empirically, but on the basis of what people believed were right. We should probably check the assertions empirically.
Any other questions, comments?
Just add what Alan said there about students and that. I mean, one big output of this endeavor will be a new generation of animal scientists that should be proficient, at least doing various levels of bioinformatics. I would be kind of, I'm quite controversial. I would say if you're in this area, you must, especially younger people, you must learn how to do some large-scale analysis. If you don't, you're not going to get to play in the genome area. And there's so many labs around that will be able to teach people how to do this kind of thing. And there are a lot of funding opportunities as well. Like I said this afternoon, there are plenty of opportunities for sending people around, and we should take advantage of them. And I'm sure all the funders in here will agree on that one.
I guess that's in part at least what the RCN is there for.
Okay, any others?
About this choice of assays, and examining what the consortia indicated, okay? It is also the issue of the resolution of these assays, which I think is actually one key thing, because, at the end, even if sequencing costs decrease, still, for example, you should produce 100 million fragments for RNA-seq, 50 million for small RNA-seq, and then for histone marks, for many of them, you don't actually reach saturation, but so you should have, let's say, and also they should be running duplicate. So anyway, it's kind of, this is the other component to consider. If you have enough critical big data set, that then you can say, okay, let's see what is really important to do, but it's very important to assure that everybody follows these standards, at least in the first place. Otherwise, how you can reason about that?
I think we have a reasonable amount of existing data, and also the pilot projects that are underway will give us species-specific context for those sorts of questions.
Any others? Okay, let's thank these guys for summarizing. Great job. With that, I'm going to turn it back over to Chris, who's got some announcements for us.
Thank you. I am not going to keep you long. Just a couple of follow-ups, just so you know what we've got planned for keeping track of what just happened in the last couple of days. We'll be putting up all the slides of all the talks on www.phenome.org, and you can look for them there. We'll have recordings, as Julian has been faithfully doing for the last day and a half, of all the full session talks as well. Both of those we should have up by the end of next week. If you have any comments about the meeting, you can certainly send it to fang@iastate.edu, which is our our clearinghouse for the emails back to the Fang website. I just wanted to end a little bit by thanking a number of groups. I'd first of all like to thank the administrators that gave us the welcome and for showing interest in Fang. It was Undersecretary Wateki was here yesterday, and Dr. Ramaswamy and Dr. Miller, I think, is still here. It was really, really great to have them here. I'd also like to thank the sponsors again, National Science Foundation and NIFA for giving competitive grants for this, as well as the USDA coordinators for funding. Illumina provided support as well, and so did our university. So I'd like to thank them again for supporting this conference. I guess I'd also like to thank the agencies that attended. At least I've never been in a conference before where the agencies came and had several meetings themselves, as well as participated in the conference itself. I think that shows incredible interest from the agency point of view, and they gave us a lot of very helpful comments. And so I think that was really, really special for me to see that we really filled our hour that we asked them to talk as well. I'd like to also thank all of the people that attended. It's amazing to see that there's about 85 of us still in the room at the end of the day on Thursday. We had everyone but one name tag taken, and the only one that wasn't has the stomach flu back in Arkansas with his family. So everyone came. So I really appreciate seeing that. Don't forget that the next workshop for Fang is January 11th in San Diego. So, and with that, thanks again for coming. And should say I negotiated that the bar opens at 5:45 instead of 6, but dinner will be at 7. Both of those events are in the Great Hall. Okay. Yes, Janice Borman as well. She was at the registration desk for yesterday and today as well. So thanks to her. Yep, and I thank Julianne Hu for taking care of all the video for us. All right, thanks very much.
I think I to thank Chris Taco to make so much of the effort to organize this.
Go have a drink.